All right, here we are at chapter 14. In this chapter, we're going to be learning about nucleotides and nucleic acids. Our objectives: to learn about DNA and RNA and stuff about that. And you got a chapter 14 take-home quiz. All right, when we talk about nucleic acids, there are a couple of biggies that always get included. One of them is deoxyribonucleic acid, and that's what we know as DNA. The other one is ribonucleic acid, which we just routinely call RNA. Now, like our fats and our proteins, these are biomolecules. They're going to be used to transmit genetic information. So that little double helix with those little base pairs in between, it contains the blueprint that is how to make another one of you. Encoded within these different nucleic acids are our genes, which are segments of DNA with instructions for making a specific protein. Mostly proteins, but there can be other things as well. We're going to start our discussion talking about nucleotides. So what's a nucleotide? It's a monomer that's going to make up the nucleic acids. Now these are going to be molecules that have different functional groups. And the thing about this type of molecule is that it is something that you're going to see repeatedly in nature. So if you were to go on and take a biology class, you would actually learn a fair amount about this type of a molecule. Now the functional groups that are involved in this nucleotide are going to be a sugar ring. This is your sugar ring right here, and yeah, it's like a glucose. And then you're going to have a phosphate group right there, which is going to be a phosphate and some oxygens. And then over here you're going to have some sort of a base, a nitrogenous base molecule, which could be any number of subgroups, kind of like an R group, except it's not R, it's an N. It's an N thing, because it's based upon being nitrogenous in nature. So this is the basic structure. Now believe it or not, if you know anything about ATP, you've already been introduced to this molecule in some way. Because if we were to take and add a couple more phosphate groups onto it, this would basically be the generic format of an ATP molecule. So that's what I was saying. When it comes to Mother Nature and she finds something that works, she has a tendency to repeat it throughout various systems. And this phosphate sugar nitrogenous base is one that gets repeated a lot in the body. Yep, base. Oh, yeah, let's talk about those bases. I forgot this was on this slide. So it depends upon what we're talking about. DNA has 4 bases, and RNA has 3 of the same that DNA has, but one's going to be different. So if we're talking about DNA, the 4 bases that could be up here in this blue box could be adenine, guanine, cytosine, and thymine. If we're talking about RNA, we're going to still have adenine, guanine, and cytosine, but instead of thymine, we're going to have a different base. We're going to have one called uracil. All right, what about that sugar ring? Well, I mentioned that deoxyribonucleic acid and ribonucleic acid were the full names of DNA and RNA. And if you listen to the language, one is deoxyribose and the other one is ribose. So ribonucleic acid, deoxyribonucleic acid. So what is being deoxygenated? Well, it is this right here. That's losing the oxygen. It's the second carbon on that sugar ring losing its oxygen. That's what makes it a deoxyribose. If it still has the oxygen, it's a ribose. If it doesn't have the oxygen, it is a deoxyribose. This is the sugar ring, deoxyribose, that makes up DNA. If it still has that oxygen there, that makes up the sugar ring of RNA. Now, this is a Furnose ring. Furnose ring has 5 carbons, 4 of which are in the ring. Here's carbon number 1, carbon number 2, carbon number 3, carbon number 4. These 4 are in the ring. 5th carbon forms this little tail up here. And the same thing is true over here. The only difference between these 2 rings is either it contains the oxygen here off that 2nd carbon or it doesn't. Otherwise, they're identical. So the carbons are going to be labeled according to their number. And this carbon is going to be bonded to the nitrogen at the primary carbon, which is also known as the anomeric carbon. And it's going to be in what's known as a beta nitrogen glycosidic bond. Now beta, this beta right there, tells us that the orientation of the bond coming off this first carbon is going to be in the up position. And that what it's going to be bonded to is a nitrogen in that base, that nitrogenous base. So it is a union between the nitrogen of that base and the sugar ring, which is why we say glyco, and that bond is going to be in the up position. It's going to occur above the ring, which is why it's beta. So that's what all of this means. It's a beta in glycosidic bond, and it's occurring right here up in the air, just like I showed on the previous slide with that base pair coming up here somewhere. All right, nitrogenous bases. These are going to be made either from pyrimidines or purines. And as we can see from this picture, a pyrimidine is going to be a hydrocarbon ring. There's a carbon and it'll have some hydrogens attached to it. Carbon with some hydrogens, a carbon. Instead of a carbon, we're going to have a nitrogen here, but we have a carbon there and we have another nitrogen right there. And we have the same thing going on here, except where we have an N, we're going to have the nitrogen instead of a carbon, and this is what makes up these two rings. So the pyrimidine is a single aromatic ring, and the purine is going to be a double ring, and that's what our nitrogenous bases are made of. There's going to be some things that are added to them that will give these various nitrogenous bases their identity. So I believe we can take a look at those. So if we look at cytosine, thymine, and uracil, we can see that these are the pyrimidines. These are made up of a single ring. And if we just note some of the differences, cytosine just has this amine group off of it. Thymine has an oxygen, but then it has this methyl group right there. And uracil, pretty simple, just has an oxygen group. Uracil is what we would find with RNA. Thymine is what we would find with DNA. Cytosine we would find with both. That leaves us with adenine and guanine, and these are going to be purines, double rings, double ring. And if we look at what's happening here, here we have an amine group coming off of this carbon, and analogously over here we have a doubly bonded oxygen, but then we have an amine group coming off over here. Now the way that these guys are going to pair up is going to be kind of dependent upon whether it's a double ring or a single ring. Because when we're talking about pairing up these base pairs, this is what we're talking about pairing up. So that when our two strands of DNA are joined together, what's going to really happen here, the essence of it, is that you're going to have a purine, which is a double ring structure, binding up with a single ring structure. So in the case of adenine, it always couples up with thymine. In the case of guanine, it always couples up with cytosine. That's with DNA. And why is that the case? Well, part of it has to do with real estate. If we had two pyrimidines trying to, two purines trying to bond together, we would end up kind of like knocking heads in places because of how these structures are going to be exposed. And these two guys wouldn't be able to bond, like two of these wouldn't be able to bond together in the right orientation. But this guy can adequately bond with this guy, and it has to do with this oxygen binding with this hydrogen, forming a hydrogen bond. The same thing is true with this guanine. Guanine is going to bind with cytosine, and this oxygen right here is going to come over here and bond with that hydrogen, creating a hydrogen bond. So it is, as we're going to take a look at in just a second, it's the hydrogen bonding between these base pairs that holds the two strands of DNA together. And the reason why you have a purine and a pyrimidine coming together has to do with basically real estate. You can't get two big old clunky rings forming a union. It's easier to get a clunky double ring forming a union with a single ring. Yeah. So we've talked about those nitrogenous bases. Let's talk about the other end of this molecule, the phosphate group. So the phosphate group is going to be what joins to the sugar ring at the 5th carbon. So here in this picture we see our nitrogenous base right here, which happens to be adenine. And then at the 5th carbon, 1, 2, 3, 4, 5, we see that phosphate group joining in right there. Now, if there's no phosphate group attached to that 5th carbon, instead of calling this a nucleotide, it would be called a nucleoside. So you can see this guy up here has the phosphate, he would be the tide group, and this guy has no phosphate, he would be the side, nucleoside. All right. So why is that important to know about the phosphate groups? Well, because it's the phosphate groups that are going to string those sugar rings, going to join with the sugar rings and create what's known as the phosphate sugar backbone of DNA. So if we look at this image right here, what we've got going on is starting up here at our 5th carbon, we have a phosphate group. But then bonded to the 3rd carbon of this ring is another phosphate group of this ring. This phosphate is attached to the 5th carbon, but the phosphate group from the ring below it is attached to the 3rd carbon. So this alternating sugar ring, phosphate group, sugar ring, phosphate group, sugar ring, phosphate group is what creates for us as what's known as that sugar phosphate backbone. Now these nucleotides are going to be strung together in a head-to-tail fashion very similar to what we saw with amino acids. And the way that we note this head-to-tail fashion, in the amino acids we talked about the N-terminus and the C-terminus. We don't have that here. In this case we're going to be talking about the 3' to 5' direction. So what we see here is this is our 3', this is our 5'. And so it has to do with orienting from the 5' all the way down to the 3', or vice versa. So we could almost think of this as like our N-terminus, and this is our C-terminus if we were talking about an amino acid. Hopefully that analogy makes sense, because hopefully you understood it in the previous chapter. Where these phosphates bond to, here we go, where these phosphates bond here to this carbon, that's known as a phosphate ester bond. Not that big of a deal. I just want you to understand that it is the phosphate sugar alternating arrangement, that head to tail fashion between the phosphate groups and the sugar rings, that's creating that sugar phosphate backbone and is going to lead to this chain structure of our DNA. That's what I really want you to know. And here's that picture enlarged so you can kind of see what's going on one more time. So here we have adenine, right here adenine, and it has a sugar ring, 1, 2, 3, 4, 5, and off this 5th carbon is a phosphate group, but attached to its 3rd carbon is the next nucleotide in the series, which would be cytosine. I know that because of the C. And it's attached to the previous nucleotide by way of its phosphate group attaching to that 3rd carbon. Here we have the 5' carbon. But then attached to cytosine's 3rd carbon is another phosphate group of another nitrogenous base known as guanine. So it's this alternating fashion that creates this sugar phosphate backbone. And when you look at the double helix of DNA, those strands are going to be what make up, are going to be comprised of the sugar phosphate backbone. The rungs, the things that are in the middle that go from one strand to another, are these guys right here. These guys right here. These guys right there. All right, so I was just talking a minute ago about these strands right here. These strands are the sugar phosphate backbone, and what's coming off of the sugar phosphate backbone are the various nitrogenous bases. So here is thymine, here's cytosine, and guanine, and adenine. All right. So DNA is made up of 2 strands of linear polynucleotides. So linear means that they're side by side. That's basically what it's saying. And they're going to have directionality, and we can see that in this picture. We have 5' to 3', and then this strand right here is going 5' to 3', but it's going in this direction. So this one's going 5' to 3' in the down direction. This other one is going 5' to 3' in the up direction. These strands are said to be anti-parallel, which means that the 5' to 3' ends are oriented in opposite directions. And I just pointed that out on this picture. So if we were to say that we had, oops, let me back up. If we were to say that we had a strand that was parallel, we would say that we would have our A and our B in the same direction. Everybody's going in, head to tail in the same direction, and that would be what we call parallel. Anti-parallel is going to be when we have, let's say, our A end starting down here at the left, but on the other one we have the A end on the right. So they're going in opposite directions, and that's what's called anti-parallel. Anti-parallel is what our DNA is, the directionality of our two DNA strands. They're basically going at it in opposite directions. The phosphate sugar backbone, as a result of that, is going to have chirality. Thank goodness we don't really have to pay attention to what that is. But you will need to know this. The phosphate sugar backbone is polar, mostly thanks to those phosphate groups which carry with it a negative charge. And as a result of having that polarity, that negative charge, is going to make it hydrophilic. Hydro meaning water, philic meaning loving. So that's actually kind of good, because what does this exist in? A water environment, our bodies. So if it's going to have a polar region and a nonpolar region, we want that polar region sticking on the outside so that it plays well in the water environment. So overall, we would say that DNA has a negative charge, and it's mostly due to those phosphate groups that are making up the sugar phosphate backbone. All right, so the base pairs are what are coming off of the sugar phosphate backbone, and they're pairing up according to this combination: adenine, thymine, guanine, and cytosine. And we can see that's what this A and the T stand for, and the C and the G. G and the C, rather. So the base pairs are formed when the nitrogen of one strand bonds with 2 or 3 hydrogens of the neighboring strand. So when one DNA base pair finds an analogous pair, or a reciprocal pair, maybe I should say, that's how that works. And this is how they always couple up. And part of it has to do with the real estate thing. Like I said, taking up a lot of space, not taking up so much space, and look at how all of that happens to line up. All those hydrogen bonds, boy, isn't life grand. Same thing here, isn't life grand? So, and it's these little hydrogen bonds, these little attractions between the hydrogens of one strand and the oxygens and nitrogens of another strand that's holding the two strands of DNA together. That's an amazing thing. All right, I already said this, the DNA is going to couple up according to the nitrogenous pairs, adenine and thymine, and cytosine and guanine. So where one strand has adenine, it will couple up with thymine on the neighboring strand. And where one strand has cytosine, it will couple up with guanine on the neighboring strand. So that's what we mean by that. And the picture kind of tells you that as well. So if you look right here, we have thymine, and it's coming together with adenine. And here we have cytosine, and it's coming together with guanine. And these little dotted lines, those are your hydrogen bonds occurring between the base pairs. All right, so DNA is held in the helical shape, not only by those hydrogen bonds occurring between nitrogen base pairs, but also by something called base stacking. And that has to do with those nitrogenous rings. So the way the rings are oriented, it will create part of that twist that we see that's characteristic of the DNA double strands. And although these forces are fairly significant, they can be disrupted. And that's going to be necessary when DNA gets replicated, because in order to replicate the DNA, we have to pull the strands apart and then copy the two strands. So if we're going to make another cell, like through mitosis, or if we want to make a gamete, I'll get it out in just a second, such as a sperm or an egg, we have to copy the strands of DNA. And in order to do that, we have to untwist them, and then we have to separate them. So the forces are significant enough to hold the two strands together so that they maintain integrity, but they're not so strongly held together that you can't pull them apart. It requires an enzyme to do that, but it can be done. All right, so the DNA strands are going to be coiled around these little proteins that are called histones. And histones are going to act much like spools for thread, where the histone is the spool and the thread is the DNA. And histones are going to have a positive charge, and that works out really well because what kind of charge does the DNA strand have? It has a negative charge. Thanks, phosphate groups, for that negative charge. It allows me to hang on to a histone so much easier. So when you have the combination of DNA wrapped around a histone, we call that a nucleosome. And then nucleosomes are going to further coil in an act called supercoiling. So what's a supercoil? It's a coiled coil. And it's going to form a larger structure known as our chromosome. This is a really good picture because it gives you an idea of what's happening. So if we start right down here, we have a strand of DNA and another strand of DNA, and they're twisting around each other. And as if that's not enough, they're going to find one of these little histones, and they're going to twist around that, much like thread twists around a spool. And then once you get enough of these are going to twist around together. And then this twisted thing is going to supercoil into this coiled coil, supercoil, and then it's going to end up with this X-shaped spaghetti sort of craziness right here, which we call a chromosome. And a chromosome is what we see right before DNA gets ready to replicate. Now, if you're going to take biology, this is going to be something that you're going to need to know. But DNA normally exists in the nucleus. It tends to be a little bit more relaxed. And it's only once the DNA has replicated that we'll see it tightly coiled like this, because it has to move through the duplication process and then the separation process in this fashion. And then once it gets to its new home cell, the new cell that it's going to be as designated as its new home, and the nuclear envelope reestablishes, then it will start to relax back into kind of something that's not as easily visible. But the whole point is, by the time you see it like this, this is what we see right before DNA gets ready to separate, and is tightly coiled like this when in the replication process. Normally it just ends up being kind of like strandy, but when we see this X sort of thing, then we know the chromosomes are getting ready to do something. Good news is that's not part of the chapter. I just kind of threw that in there. All right, what's a genome? Well, the genome is a complete sequence of DNA nucleotides, that is, those base pairs, distributed over 46 chromosomes. 2% of the genome is going to code for proteins, and the rest is going to be kind of information about, you know, hair color, eye color, skin color, shape of your toes, things like that. So a gene, then, is a DNA segment that contains instructions for protein production. Now, humans have about 30,000 genes, and the role of most of those is unknown. Now, feel as you may about all the sorts of things that they're doing with genes these days, I'm still a little bit uncertain about a lot of the gene technology, because I don't think we know exactly what most of those things are doing. So I got my opinions about that, but this says a lot. And there may have been an update since this book was written, and it may be that we now know a little bit more about those 30,000 genes, but we probably don't know enough about 29,995 of them, which would be about the level I'd want it to be at before I started letting people mess around with my genome. So, all right. So what happens during DNA replication? Well, once DNA is going to be replicated, the two DNA parent strands separate. And what we mean by parent are the strands that you've got. So the strands that you got are going to serve as the template for the ones that are going to be made. So we call them parent, and the ones that get made are called the daughter strands. So that's pretty common language that you see in the health sciences. So when you make a replication of something that's existing, the thing that is existing is called the parent, and the thing that gets created as a result of that thing that was existing is called the daughter. So each parent strand is going to serve as a template for the complementary daughter strand. It's also known just as the daughter strand. It's called complementary in this case because of that whole base pair business. So what's going to end up happening is that the two strands are going to separate about like that. And then we're going to copy the strands. And if you look closely, where we have a G, we're going to put a C. Where we have an A, we're going to put a T. Where we have a C, we're going to put G. So we're going to see that complementary strand being created. It's going to be the opposite of what we have. And if you noticed how I did the graphics on this, for the top strand I started from the right going to the left, and for the bottom strand I started going from the left and going to the right. And there's a reason for that. And we're going to talk about that in a little bit. Now, DNA polymerase, we refer to it as a singular enzyme here, but actually it's a group of enzymes that are doing a bunch of different things in order to help this DNA strand replicate. But generically, we could think of DNA polymerase or the group of DNA polymerases as the enzymes that help create the sugar phosphate backbone that will allow those nucleotides to create the base pairs. So basically what we have to do is we have to call in the appropriate base pair and hold that in position and then bring in the next base based on the pairing, and then knit those two together, and then call in the next one, and then knit those two together. Sounds a little bit like DNA about amino acid chain assemblage, and that's true, it is. Because in order to, I'm going to start over here at the right, over here, we have to call in the C, and we know that it's going to be C because of this G, and we have to hold this in place in order to bring in the next one, which is the T, and we know it's going to be T because this one is A. And then we have to create this bond right here, because all of this is the sugar phosphate backbone. And these are among the things that DNA polymerases are doing for us. Fortunately, you don't need to know any more than that. We can keep it just that simple. All right, so the parent strands are going to be replicated in one direction. And the strand that's being created wants to start at the beginning and be reproduced all the way to the end. And with DNA, the starting position is at the 5'. So the daughter strand is going to be made anti-parallel in the anti-parallel direction of the parent strands, which means that the daughter strand's 5' is going to start at the parent's 3'. So this strand right here is going to serve as the parent strand for the daughter strand that gets made. And this daughter strand will be made just like this. So this daughter strand is being made 5' to 3' from the daughter strand perspective. But what about this strand down here? This strand down here, we're starting at the parent's 5' end, which means that we're starting at the daughter's 3' end. That would be the end. We can't start making from the end and going from the reverse, because the daughter wants to be made 5' to 3'. So what happens with this strand is that we make it in little sections, because if we start like say right here, then we can make a little section going from the daughter's 5' end to the 3' end, the daughter's 5' to the daughter's 3'. So we make a little section, and then we make another little section, and then we have to knit those two little sections together. So that's how the two strands of DNA are replicated. One gets replicated continuously, and the other one gets replicated in little sections. Now, as we unfurl DNA, part of it's going to be held together still, and part of it's going to be separated so it can be replicated. And then once we're done replicating it, it'll be knitted back together. Now where it stays together but has been separated, where it's together yet separate, we call that the replication fork. Good to know. Alright, so I talked about these two strands being replicated a moment ago. And this upper strand that's being made from the daughter's 5' to the 3', following the parent's 3' to 5', this one gets assembled continuously, and we call this strand the leading strand. Now the one here on the bottom is the one that's going to be made in these short fragments, and we call that the lagging strand. And that's the one that gets made a little bit at a time. As the DNA gets separated and undone, then it gets to create the next little section. It takes a little bit longer, but it's still being made in the 5' to 3' end according to the daughter's perspective. Now, I talked a little bit about DNA polymerases. Now, DNA polymerases are going to be working on that daughter strand, and it's going to be doing things like calling in the right base pair and making sure they get knitted together correctly. And there's a whole bunch of those, but we're just generically calling it the DNA polymerase. It's catalyzing the daughter strands, assembling those daughter strands. Now, the helicase is going to be the enzyme that uncoils the parent strand. And why do we need to uncoil the parent strand? Because the next thing we need to do is separate it. So in order to do that, we can't separate it while it's still all twisted up. We have to untwist it, because remember, it's a supercoiled coil. So we got to undo it so that we can get to it. DNA ligase is going to join the fragments of the lagging strand so that it's continuous. So DNA polymerase is going to bring in those nucleotides and make sure we have the right base pair in position, and then knit that sugar phosphate backbone together. And then it's going to be DNA ligase that joins the fragments of the lagging strand together. So these are some of the players. There's a lot of players that are needed to make this happen. These are just some of them. Some of the more interesting players in the formation of the DNA daughter strand. DNA replication occurs at the rate of 100 nucleotides per second. And the error rate is only 1 in every 10,000 nucleotides. That's pretty good. That's real good, actually, which explains why, for the most part, we turn out okay. All right, let's talk about central dogma. So central dogma is language that you're going to encounter in various fields of scientific study. And what central dogma more or less tells you, and a lot of people are going to frown when I say this, is that what central dogma tells you is that this is our best understanding of what we think is happening because we don't really know for certain. So what we're talking about here on this particular slide is the central dogma related to how we go from DNA to protein. This is our current understanding of it. This is our best model of it. So let's start by talking about the 3 different types of ribonucleic acid that are involved in producing a protein from the recipe contained in our DNA. So we have one guy that's known as the ribosomal RNA. Can you guess where he's found? Hopefully you said a ribosome, because that is exactly where he's found, within a ribosome. Then we have something that's known as messenger RNA, and I'll talk about that in the next slide. It'll make more sense if you've got to have a picture that goes with it. And then the same thing for transfer RNA. So let's get into this part of the discussion. All right. So very wordy slide here, bear with me, and then we're going to look at some pictures that will make it so much clearer. So the messenger RNA is going to be what's created from copying a section of DNA. So if we think about DNA as a big recipe book on how to make proteins, hopefully that puts that into context. So if you needed to make a protein for something like, I don't know, maybe some hair, you would go to the section of DNA that talks about making proteins for hair, and you'd copy it. And the way that you would copy it would be to make this little section known as a messenger RNA. The only thing that would be included in that is the recipe for making hair protein. Now when we do that, when we're in the process of making that messenger RNA from a section of our DNA, we call that act transcription. And much like replicating the entire strand of DNA, if we're going to copy just a section of it, we copy it very much like we would copy an entire strand. It goes from the 5' to the 3' direction. Now where is DNA located? DNA is found in the cell's nucleus, which means that our messenger RNA is being made in the nucleus. It means that transcription, which is the creation of messenger RNA, is happening in the cell's nucleus. And of course it takes an enzyme to happen, and we would call that an RNA polymerase. So that's a little bit about messenger RNA. Messenger RNA is a sequence copied from DNA. It's just a single strand, unlike the DNA, which is 2 strands. Our RNA is just a single strand. Now tRNA is, it says that it reads the messenger RNA and produces the amino acids. I'm going to phrase that a little bit differently. What tRNA is it's going to be the carrier for amino acids. So we could think about these little units of ribonucleic acid latching onto an amino acid that got put into the bloodstream by way of the diet. And it has on it base pairs because it has to communicate with that messenger RNA strand. And the way it does that is much like the two strands of our DNA. It has to have base pair matching. So our transfer RNA is going to have attached to it an amino acid. And it's going to recognize that it needs to bring in that amino acid because of how that messenger RNA gets read. Now, who's reading that messenger RNA? Another ribonucleic acid called rRNA, ribosomal ribonucleic acid. It's going to read the messenger RNA, and we can think of it more like calling in a transfer RNA. That's what the T stands for in tRNA, transfer RNA. And it's going to call in a transfer RNA with an amino acid, and then it's going to assemble the amino acids together to form that growing amino acid chain, which we call a peptide, until it becomes big enough to be called a protein. So let's put all of this together in pictures, and hopefully it will make more sense. All right. Yeah, this little bit too. Forgot about this. So how do we get amino acids from messenger RNA? Well, those base pairs, that ACGT business that we're talking about, except it's not the T in ribonucleic acid, it's a U. It reads those 3 at a time. And every 3 base pairs describes a particular type of amino acid. So when we look at the 3 base pairs together, we would call that a codon. So the codon is the recipe. It's a recipe of the 3 nucleotides that tell us which amino acid is next in the sequence of making our protein. So if this is our DNA template, we've copied our messenger RNA, looks like that. So we can see we have UUA and CAG and GGC. Each one of those 3 base pairs, AAU, that's a codon, and it means I'm looking for a particular amino acid. CAC is another codon. It's a different amino acid. GGC is a codon looking for yet another amino acid. So that's how our ribosomal RNA knows which amino acid to call for from the transfer RNA. It's by reading the messenger RNA nucleotides 3 at a time in the form of a codon. I don't expect you to know which codon codes for which protein, but this is or which amino acid, but this is an example of that. There are charts. You can go look this up, and I believe that UUA does code for leucine, and the other 2 are in place. But I don't expect you to know that. That's the good news. All right, I believe this is a picture that might be coming from your book that talks about what's happening here. So what we have, I'm going to get you oriented to this picture because you'll see a lot of pictures like this. Here we have our messenger RNA. This came from copying the DNA in the nucleus in an act called transcription. It leaves the nucleus and comes out into the cell, and it's looking for a ribosome, which contains this sort of beigey green colored unit known as a ribosomal RNA. And this ribosomal RNA is going to read this messenger RNA like a ticker tape. If you happen to know what that is, it's a tape readout that's kind of cryptic language, and people who know how to read it know what it means. It was once used to indicate what was happening in the stock market. People had ticker tape readouts talking about what was being traded in what quantities. But in this case, this is telling us about what amino acid needs to come in next. Now you'll notice this right here. It says tRNA. These are the transfer RNA. And you'll see here that this has also nucleotides on it, and they're the complementary base pair to the one that we see here on the messenger RNA. So where there's a G on our messenger RNA, our transfer RNA is going to have a C. Where there's an A on our messenger RNA, we're going to have a U, because remember, RNA deals in U and not the T, and then vice versa. So where this is called a codon, this region is called an anticodon. I'm just including that because you might run across that language in the book, but I don't include it in the slides and I don't test over it. But we can see that this is that communication. These base pairs still end up being a communication even between the tRNA and the messenger RNA. Now attached to this tRNA is an amino acid. In this case, I want to say this is asparagine, lysine, tryptophan, and this one's bringing in phenylalanine. And they have different codes. And it's being called in, this transfer RNA is being called in based upon what's being read here in this ribosome looking at the messenger RNA. And you'll notice that there's two tRNAs sitting here. One of them is in the reading position, which is I'm calling it in because I need this one, and then there's this one right here. Now what's happening between these two is that once these two come in side by side, this ribosomal RNA is going to knit these two amino acids together. And once these two amino acids have been knitted together, it's no longer important for this amino acid here on the left to still remain attached to the transfer RNA. So this bond right here will get broken. And once that happens, this whole unit, this tRNA and this messenger RNA, will advance 3 nucleotides so that this incoming transfer RNA will now be in the reading position, and this current one right here will be in the outgoing position, and that will mean that this amino acid can be attached to that amino acid, and we'll have this growing amino acid chain. Now what's going to happen with these transfer RNAs is that once it becomes detached from the amino acid, they get to go out and find another amino acid and have it at the ready to bring in if the messenger RNA needs one. I don't believe the one pass of this in a static picture is going to be good enough, so guess what? We're going to go over it again. This is a picture from your book, and this is a beautiful picture. Goes over the same thing. I'm not going to do that here because I've got, I think, a better animation that talks about this. All right, so here we are in my very rudimentary cell, and we are going to talk about transcription, which is the copying of DNA, so that we understand where we are. This big blue area is going to be the cell interior, and the purple area is going to be the cell nucleus. And what's going to happen in the nucleus? Transcription. That's where we're going to copy DNA, and DNA is located in the nucleus. So if we bring in our DNA, there it is, DNA. And if we're going to copy it, what needs to happen? Separate it. That's right. We're going to separate our DNA, and then we're going to copy it, just a single strand. And there is our messenger RNA. Bam. There's our messenger RNA. All right. So we have just done transcription. We've copied the DNA. We have created messenger RNA. All right. So what's going to happen with that messenger RNA? Well, now it gets to leave the nucleus, and it has to go find a ribosome. And when that messenger RNA finds a ribosome, we're going to start into what's known as translation, which is the making of a protein. All right, so translation happens out in the cytoplasm of the cell because that's where we're going to find our ribosomes. We have ribosomes in a couple of places. We have ribosomes located on the rough endoplasmic reticulum, which is located conveniently right next to the nucleus. We also have some ribosomes that are just kind of free floating out in the cytoplasm. And it doesn't really matter where the ribosome is. The steps are going to be the same. So we're going to look at this as just a generic ribosome, and we'll just pretend, I don't know, we can pretend that it's an endoplasmic reticulum. It doesn't really matter. All right, so here we have our messenger RNA, and it's positioned its beginning is positioned at the ribosomal RNA, and it's in the reading position. I like to call that the reading position. So our ribosomal RNA is like a 2-unit protein structure, and it's going to be able to take a look at those nucleotide base pairs, and it's going to call in transfer RNA and make sure that it's bringing with it the right amino acid. All right, before we get started, we have to zip up our DNA. So zipping up the DNA. Oh, good. Okay. And now we can get started with our translation. So we have a UUA, and we got to find a transfer RNA that's going to correspond with that. Because remember, what we're reading here is that codon. So if we bring in the appropriate transfer RNA, bam, there it is, we can see that it has an anticodon right here, AAT. Yep. Oops, that should be a U. Whoops, mistake. Well, too late. And that attached to this one is this particular amino acid, and I don't remember what it is. All right, so now that we've called in the amino acid, the reading position is done. So this messenger RNA is going to advance within the ribosomal RNA, and the next codon will be read. All right, so we've advanced our messenger RNA within the ribosomal RNA, and we have the one that was read previously in that position waiting for the next one to be called in so that we can get those two amino acids hitched up. So let's call in another transfer RNA and its corresponding amino acid. Looks about like that. And now what's going to happen is that we're going to knit the first amino acid to the second amino acid. And we're going to release the first amino acid from that transfer RNA. All right. Now that we're done with that, we're done with that second amino acid being in the read position, and we can advance this whole thing forward. All right. So now we're going to read that next codon, and we're going to call in another transfer RNA. But the first transfer RNA, now that it's unhitched to that amino acid, is going to be free to go find another amino acid to link up with and have at the ready for when there's that next calling for this particular amino acid based upon the codon. So we're going to say bye-bye to the transfer RNA. It's going to go away. And then we're going to call in another RNA and its corresponding amino acid. And there it is. And what are we going to do with those two? We're going to knit those acids together. We're going to release the second amino acid from its transfer RNA, and we're going to advance the entire unit within the ribosomal RNA. And if my messenger RNA was long enough, I'd read the next codon and bring in another amino acid. Now, what do we have right here? Oh yeah, bye-bye transfer RNA. So what do we have right here? Well, we have these three amino acids strung together. So this is our growing amino acid chain. This is becoming a peptide. We've got a peptide right here. We've got three amino acids strung together. Eventually, we're going to have, I don't know, several hundred of these strung together in what's going to become a protein. And that is how we make proteins in the body. That's what's happening when you make some keratin or something else. It happens just like this. Happens a lot faster than we just went through it. But that's what's happening every single time. So now we understand, hopefully, understand the roles of messenger RNA. Gets created in the nucleus by copying a section of DNA. And then we create the protein in the act of translation. And that involves all three of those RNAs. We have to have the ribosomal RNA reading the messenger RNA, calling in those transfer RNA and the corresponding amino acids, and then knitting it all together. All right, what happens when things go wrong? Well, you get something called a mutation. So a genetic mutation is a permanent chemical change that happens within the nucleotide sequences. And it affects the primary structure of the protein because it's affecting just that amino acid sequence. A couple of different things can happen. You can have a substitution where one nucleotide gets substituted for another. That can happen. Or you can have what's called a frame shift, where a nucleotide gets added or deleted, which will throw the entire replication process off and can lead to something new. Sometimes the things that get created are good. Sometimes the things that get created are bad. Sometimes the things that created are good that get created can be good and bad, such as sickle cell anemia. I think I covered this on the next slide. Let's take a look. So let's talk a little bit about sickle cell anemia. Sickle cell anemia is a malformed red blood cell. Now usually red blood cells are round, and they eject their nucleus, so that makes them pliable, which is a good thing because they're flying around the body at a really rapid rate of speed, and they have to be able to squeeze down through those little capillary beds, and then rub elbows with the rest of the red blood cells, and they got to be able to have like a little bit of slip and slide. We don't want our red blood cells getting stuck somewhere. We call that a blood clot, and that's a problem. Well, the problem with having red blood cells that are shaped like a curve is that they get stuck somewhere and create kind of like a blood clot, and that creates a problem. The thing is that having that sort of a shaped red blood cell leads to some protection against malaria infection. Now, I don't remember all of the details about that, I guess is what I should say. I used to know it, I don't remember it now. But it provides some protection against malaria. So you'll see sickle cell anemia in populations that have been in subtropical areas like Africa and in Asia. So those populations have a tendency towards sickle cell anemia, and in part it's because this mutation happened and the people who had it had a tendency to survive malaria. So it got perpetuated and gets passed on in the gene. So it's a mutation that occurred, creates a problem, creates a whole host of health issues, but if you live in a subtropical area, it'll provide some adaptive advantage against malaria. Cold adaptation is another interesting genetic mutation. And we think that this one comes from maybe even Neanderthal times, or certainly some archaic human beings. And what happens here is that fat metabolism got altered somehow. And what ends up happening is the way fat gets stored and the way the fibroblast, which is one of the cells that creates scaffolding fibers for fat cells, got created, allows fat to be put down differently in the body. And as a result of that unusual fat deposition, it creates better insulation against cold, and a cold adaptation that we see in certain Inuit tribes. Now, there's a lot about this we don't know still because the genome hasn't been sampled across all populations, in particular the Inuits of Greenland. And but we do know, having done some study of the genome, that this is an unusual adaptation that has occurred in some of the Indian tribes that live in the northern climates, that they have a different way of depositing fat and harboring fat than, say, someone like me of northwestern European descent. The last one that we look at as far as genetic mutation has to do with something with our blood. Now, on our red blood cells, we can have something like 250 different types of proteins, antigenic proteins specifically. Antigenic meaning that it could elicit an immune response. So our immune systems need to be sensitive to these different types of proteins that can be on there. Now, when it comes to blood typing, the types of proteins that we're looking at are called an A type and a B type. And if you don't have A or B, we would call it an O type. Because it's the absence of A or the absence of B or the absence of both of them, so which is what leads to O-type blood. We also have something that's called Rh negative blood or Rh positive, and that has to do with a series of Rh factors. Now, if I were going to teach this in under certain circumstances, I would tell you that it has to do with an antigen D. In reality, it has to do with several antigens. One is D, the other one is C, and then there's another one called E. And if you have certain combinations of these, that can lead to something known as Rh positive or Rh negative. But there's a few people on the planet that have no D, no C, and no E. And they are so rare that there's only about 40 people alive today that have that in their blood. And we call that Rh null, not Rh negative, because they don't have any of the three indicators for the Rh factor. None of them. And it makes them really valuable because there are certain people who have all kinds of issues that would really benefit from getting a dose of Rh null blood. It would really reduce the risk of their bodies rejecting that type of blood. Of the 40 that are alive today, only 10 are currently donating blood. Chances of you and me being one, not good. I'm Rh negative, but I don't know if I'm Rh null. I have a tendency to believe that I am not. So anyway, each one of these is a genetic mutation that happened either as a nucleotide substitution or a frame shift. So, like I said, sometimes they work out to your benefit, sometimes they don't. And with that, we have concluded all the lectures in this term. Thanks for listening. Bye-bye.